Hi, thank you very, very much for the opportunity to speak here. It's wonderful to get a chance to talk about this work and also to meet so many of you over the past couple of days. So, my name is Rahul Satija. I'm a postdoc at the Broad Institute in Aviv Regev's lab. And this is really joint work with Joshua Levin's group, who's really one of our RNA-seq gurus at the Broad Institute. We do a lot of RNA-seq, but today I want to talk about some of our more recent efforts to try to expand the power of the technique to go from sort of more classical samples to more challenging samples, particularly those that we see in the clinic. So I think hopefully many of us can agree that RNA-seq is potentially a very powerful tool for measuring gene expression. So around 2008, there were a flurry of papers that came out showing that RNA-seq is not only a very highly reproducible technique, but can be applied to a wide variety of systems, and in particular, can capture new transcription and capture transcriptional events that people hadn't previously anticipated. For example, widespread transcription across the yeast genome. And so it became very clear very quickly that converting RNA to cDNA and then applying standard cDNA sequencing techniques was a very powerful tool for mapping the transcriptional landscape. But it's important to point out that in terms of mapping the transcriptional landscape, estimating gene expression is only one of the powerful things that you can do with RNA-seq. And immediately, shortly after the first papers were published, there became a next emphasis on what are some of the other things you can measure with sequencing-based technologies. And one of the most exciting things is actually not only the ability to measure expression, but also the ability to measure different splice isoforms. And it soon became possible to appreciate that between different tissues, not only did you see differential expression of genes, but also different exon inclusions, different transcript structures. And this has significantly broadened the field of post-transcriptional regulation and expanded our understanding of RNA biology. Another quite cool application of RNA-seq that my lab is, or Aviv's group is particularly interested in, is the ability to actually assemble a complete transcriptome without a reference genome sequence. So even if you don't have a reference genome sequence to map, it's possible to try to reconstruct the transcriptome entirely from short reads. This is particularly valuable for certain plant species where the genomes are in fact so repetitive that it can be quite difficult or inefficient to sequence the genome. But if what you care about is gene expression, it's possible simply to do RNA-seq and get the transcriptome just from there. So there are many, many things that we can do potentially with RNA-seq. And it's a potentially powerful tool, but there are also a multitude of challenges, particularly if we go to more complex samples. So even as recently as about a year and a half ago, if we went to the Broad Institute and said we want to do an RNA-seq experiment, they would say, okay, well, you want to start with about 1 microgram of RNA, which corresponds to about 100,000 mammalian cells or so. It's actually quite a lot of material for certain applications. So it's very challenging in the context of RNA-seq to go down to lower input, eventually approaching even single-cell RNA-seq. And this is particularly valuable if we want to analyze rare cell types or samples from tissues where there's actually a limited amount of material. But perhaps an even more common problem is the ability to actually analyze low-quality RNA-seq samples. We always wanted to take RNA samples with a RIN number of 9 or as close to perfect as possible. But what happens if you start going to the lower and lower ends, the RIN number of 5, RIN number of 6, which sounds terrible, but it's often what you see if you start going to the clinic and taking samples, for example, from a patient biopsy, where the surgeon doesn't have the ability to immediately extract RNA from the sample. Or, and one of the motivating problems for this project, what if you're looking at post-mortem samples? So the GTEx project is an international consortium to look at RNA-seq samples from more than 40 tissues and more than 1,000 patients post-mortem. And these are some of the worst, most degraded, low-quality RNA samples we can find. We want to understand, can we find protocols that will enable us to profile them with RNA-seq? So one of the major problems for all of these RNA-seq protocols is that most of the RNA in the cell is actually not mRNA. It's stuff that we don't care about. It's these ribosomal RNA peaks, which we see very clearly in a bioanalyzer trace, but actually wind up making up more than 90% of the total cellular mRNA. So all of these protocols, one of the first and most important steps for them to do is to find a way to deplete this ribosomal RNA so we don't waste all of our sequencing reads on things that we already know. Now, if you think about how do we usually do this, we can actually go back to 1972 to find some of the first efforts to actually remove ribosomal RNA. I'm sure we all do, with the poly A selection, because luckily for us, ribosomal RNAs are not polyadenylated. So this is what we call an oldie but a goodie in terms of a technique, but it's not a very good solution for low input and low quality experiments. And the reason is that a poly-A selection means that not only do you lose your ribosomal RNA, but it's an inefficient process. You also lose quite a bit of your mRNA. And when you're in a low input application, every single molecule is precious. You want to hold on to as much RNA as you can. And when you're looking at degraded RNA, you're now in a situation where maybe not every molecule actually has a full intact poly-A tail. So if you do a poly-A selection and the poly-A tail isn't present, you're going to throw away a lot of the stuff that you care about as well. So we need non-poly-A selection to deplete ribosomal RNA is a key challenge for both low input and low quality protocols. And there's certainly not a lack of availability of techniques, as our spam folders will certainly show. There are plenty of new RNA-seq kits arising all the time. And so we wanted to understand, which ones can we use? And so we started the way that we usually start things, which was to send an email around and say, has anyone tried this? And this was a disaster, because inevitably people had tried a variety of techniques, but they might have tried it on a different sample. Maybe it was RNA from primary cells as opposed to cell culture. Or maybe even if it was the same system, they would have sequenced their samples to a vastly different depth, which made direct comparison quite difficult. And often the actual bioinformatic approaches to evaluate the quality of the data varied significantly between one lab and the next. So it became impossible to actually compare protocols and to see on the same samples what works and what doesn't work. We needed a systematic approach to identify the strengths and weaknesses of every method that we tested. I'm going to talk you through our approach for doing that for 5 different protocols. But I should mention that this is hopefully something we can do broadly for a wide variety of RNA-seq techniques, including new ones that are coming out all the time. So the first thing we wanted was a sample that we could uniformly profile with across all of the different protocols. We wanted something that was reproducible and easily available. So we started with K562 RNA. This is maybe not the most interesting biologically, but we can buy it off the shelf. And we started with 1 microgram aliquots. So now every one of our protocols gets the exact same input, which is good. Now, of course, K562 RNA is both abundant and high quality, so that's not really going to help us profile harder techniques, but we can simulate a low-input RNA-seq experiment very easily just by diluting down our RNA. We go down to 1 nanogram for our low-input experiments, which corresponds to about the input of about 100 cells. And we can very easily simulate low-quality RNA simply by throwing our RNA in on a heat block at 85 degrees with some fragmentation buffer for a few minutes, at least starting to simulate the degradation process, although I'll also talk about how we actually look at this in the context of human samples. So now we have a reproducible low-input and low-quality inputs. Now we can go ahead and construct our libraries using all the techniques that I'll tell you about in a minute. But another problem becomes, well, how do we know which one is better? What's the right answer? And we don't necessarily know that. So what we chose to do was to actually create a gold standard library. We made 2 gold standard controls. One was to do a poly-A selection from a large amount of K562 RNA, as you would expect. But the other was we actually chose to just sequence a total RNA library. We didn't do any ribosomal depletion. We reasoned that if we want to understand the effect of ribosomal depletion, we're going to have to bite the bullet and make a library without affecting the sample at all. So we then went through and sequenced all of these samples to 75 million reads. We did 101 base pair paired-end sequencing. I should just point out, though, that for our total RNA library, we actually had to sequence 10 times as deeply because 90% of our reads were ribosomal RNA. So we just bit the bullet and generated that gold standard data set doing 750 million reads for a total RNA library. Now we have a data set that enables us to systematically compare our different protocols based on a variety of metrics. Certainly we're very interested in quality metrics, for example, the actual efficiency of the ribosomal RNA depletion. And then even if the reads don't map to ribosomal RNA, where are they actually mapping through the genome? Of course, we want to have high mapping rates. We'd like our protocols to have a good evenness of coverage, because if we're interested in SNPs or splicing events on the 5' or the 3' end of the gene, we want to make sure that the protocol is able to capture all parts of the transcript. And lastly, of course, maybe what we care about most is, are we able to do faithful and accurate expression profiling using each of these RNA-seq protocols? So let me just quickly walk you through the different protocols that we tested for this paper. The first protocol that we tried for low-quality RNA was to use a DSN approach for depleting ribosomal RNA. What this does is it actually denatures cDNA, so now you have 2 strands, and then partially reanneals the sample. And the idea is that abundant RNAs are going to anneal more quickly than lowly abundant RNAs. And then you can use this DSN nuclease to actually chew up and degrade your highly abundant reannealed samples. So the DSN protocol doesn't actually specifically deplete ribosomal RNAs, but it depletes highly abundant RNAs. And since ribosomals are so abundant, you get very efficient ribosomal depletion. Another option, which we actually were trying for the first time, was a protocol that uses RNase-H. So what this does is it very selectively depletes ribosomal RNA by introducing custom DNA oligos, which we synthesize, 50 base pair oligos, that are complementary to the ribosomal RNA. So these form an RNA-DNA hybrid, just through hybridization, and then we use RNase H to actually specifically cleave only this RNA-DNA hybrid. So we specifically deplete only the ribosomal RNA, leaving us with hopefully the remainder of mRNAs. Another option is to use ribo0. This is sort of a similar option. It's again doing a very targeted ribosomal RNA depletion using RNA probes now. And it's using a commercially based beads-based reagent. We also tested the NuGen. This was the Ovation V2 kit. The NuGen is actually an interesting. We were excited about this because actually it allows us to profile both low quality and low input samples. It's the only protocol we tested on both low quality and low input. And the way that NuGen works is it actually doesn't use PCR, or at least this kit works. It doesn't use PCR in the library prep. It actually uses a linear amplification method called, I think it's single primer isothermal amplification. And NuGen has found that that strategy actually preferentially amplifies mRNA rather than ribosomal RNA, which is how you get your depletion. And finally, we tested the CloneTech Smarter approach. The way that CloneTech Smarter depletes ribosomal RNA is to use an oligo-DT priming scheme. And it actually synthesizes full-length cDNA. And the way that it amplifies full-length cDNA is at the end of the RT reaction, which reverse transcribes the whole RNA molecule, you get this 3-base pair addition, a template switch that enables you to ligate on another adapter prior to doing PCR and to generate full-length cDNA libraries. So after we generated all of these different libraries, we wanted to have a uniform computational analysis pipeline. I'll walk you through each of these steps in more detail during the presentation, but I just want to point out that the very first thing we want to do, of course, is we want to find out how many of our reads are mapping to a ribosomal RNA index, a custom index that we built specifically to align for ribosomal RNA. We then remove these reads, and then we do 2 separate alignments, and we have to do, unfortunately, 2 separate alignments. First, we do a genomic alignment using TopHat, because some metrics, for example, to understand how many reads map to introns, you actually have to map to the genome. But for other metrics, particularly for gene expression, what we actually want to do is we want to map directly to a transcriptome. And we can do this for human samples because, of course, the human transcriptome is very, very well annotated. So we do a separate genomic alignment with TopHat and a separate transcriptomic alignment with Bowtie. And the very important point to make is that in order to compare all of these different libraries, we actually downsample each of the experiments to have the exact same number of mapped reads, both for the genomic alignments and separately for the transcriptomic alignments. And this means that we have equivalent sequencing depth across all of our libraries in order to be able to do direct comparisons. So how do things actually wind up looking? I'll start walking you through one test at a time. The first thing we wanted to do was say, how efficient are these different libraries at actually doing ribosomal depletion? I think I've gotten some interesting library technique names here. But I'll point out, you can see, at least from the bar graph, that certainly in our control total RNA-seq sample, there's quite a lot of ribosomal RNA. In fact, more than 90% of the reads map to ribosomal RNA, which is exactly what we expected. That's why we sequenced that library quite a bit deeper. However, this is actually the DSN-like protocol here in RNase H had extremely, extremely efficient ribosomal depletion. We were very pleased to see that, even though we weren't using a poly-A-based method. Also, the Smarter technique, which actually does use poly-DT priming, had very efficient ribosomal depletion, as did a standard poly-A selection. We did notice that the NuGen technique in the ribo0 did clearly deplete ribosomal RNA, but weren't quite as efficient as some of the other metrics. So then we wanted to ask, okay, well, even if we remove the ribosomal RNA, where do the rest of the reads map? And so what you're looking at here is a stacked bar plot. The solid colors correspond to reads which map to exons, which map directly onto RNAs. The striped bars correspond to reads which map to intronic or introgenic sequences, and the white bars map to our reads which didn't map at all. So you can see that all of these protocols have about an 80% genomic alignment rate. However, only a couple of protocols, for example, the poly-A selection and the Smarter protocol, actually had more than 50% of reads mapping to mRNA. So a lot of our reads were being wasted on either intronic or introgenic sequences. And this was actually a little bit disconcerting until, of course, we realized that when you're not doing a poly-A selection, as we're doing for all of these low-quality libraries, you're also sequencing not fully mature RNAs. So you were also sequencing RNAs where the introns haven't been spliced out yet. And that's why we're seeing for all of these a little bit lower percentage of reads which are actually mapping to exons. So it's not the end of the world, but it is true that methods which are somehow efficiently using the oligo-DT are enriching for mature RNAs, which might be something that's useful for an experiment. Another metric that we were very concerned about, this is just an introductory slide, I'll go into this in more detail, of course, is what's our evenness of coverage for each of these libraries. So what I'm showing you here is for each library, how do the reads tend to distribute across the transcript? Now, obviously, what we would want ideally is to have a nice flat line here, which implies that we have perfect coverage, of course, over the entire transcript. Things never look this good. We always see a dip of coverage in the 5' and the 3' ends. But for some of the libraries, you can start to get a sense that this is particularly bad. For example, this red library here has a very, very strong 3' bias. And so the metric that I'm showing you here is actually the percentage of genes where we fully cover either the 5' or the 3' end. This winds up being a very intuitive and accurate metric for actually comparing transcript coverage. But I should point out that when you do this, it's essential that you have equal read depth across each of your libraries, because otherwise, of course, libraries with higher sequencing depth will have higher coverage. And so what we can see is actually, there's actually quite a big difference from library to library in terms of our evenness of coverage. We see that the RNA-seq library and the ribo0 is this pink, which is getting screened out here, and DSN light is this yellow bar. The RNase-H and the ribo0 methods actually have very high coverage of both 5' and 3' ends. However, a couple of the libraries, particularly the DSN and the Nugen, showed a much stronger level of 3' bias. Now, one of the things we wanted to understand was, is this true for all transcripts? Or is it just the case that we see the strongest levels of bias only maybe for very, very long transcripts? So what we did is we split up the analysis to bin genes based on not only their, the library, but of course based on their transcript lengths. So now we can see that for all of the libraries, actually, we get worse and worse and worse 5' depletion as we go to longer and longer libraries, which I guess makes some sort of sense. As you go along the transcript and you get longer libraries, it's harder and harder to cover the 5' end. But we actually see that the DSN light and Nugen protocols actually have this sort of low relative 5' coverage for all of the different transcript lengths. So it's not just restricted to a single bin of transcript length. Now, perhaps the metric we care almost most about is what's the accuracy of quantification? How well are we doing in terms of profiling gene expression? And here, it's very valuable to have on the x-axis our real gold standard control, our total RNA-seq library, in which we did no ribosomal depletion whatsoever. So we've really done minimal manipulation of the sample, and that gives us a gold standard of expression values. So you can see that some protocols are going to correlate very, very nicely with the gold standard. Other protocols are going to correlate quite noisily. And the metric that we're going to use is simply a Pearson correlation of log-transformed FPKM values to see how well we're agreeing with the gold standard. And what you can see right away, again, is that there's quite a big variation in how well we're actually agreeing in terms of expression quantification. Again, the RNase-H and ribo0 protocols performed extremely well. As we would expect, also poly-A controls performed quite well. And in general, all of our low input libraries had much lower correlations than our low quality libraries. And that really makes sense. Obviously, RNA-seq becomes much, much more challenging once we get to low inputs. But we were encouraged that we were actually getting correlations of above 0.95 to 0.97, even for our low quality fragmented libraries. So again, we saw that RNase-H and our ribo0 had the strongest agreement across all of our different samples. One other question we wanted to ask was, again, is this true for all genes? Or do certain technical qualities of different genes, for example, the GC content, actually influence the agreement between the library prep and the gold standard? So again, we bin genes based on their GC content and then calculated within a bin what's the correlation with the gold standard. You can see that some methods, for example, RNase-H and ribo0, display extremely consistent correlations regardless of the GC content of the gene. However, there were a couple of libraries that actually had a very, very low correlation once we went to genes with very high GC content. This actually makes sense. For the DSN light, you remember that what's actually going on here is you're taking advantage of the fact that abundant transcripts happen to re-anneal faster than lowly abundant transcripts. But of course, transcripts with high GC content also tend to re-anneal faster. And so as a result, you end up artificially depleting those transcripts in our library. And for the smarter method where we obtain the exact same thing, we think this is because in the long full-length amplification process, we're actually preferentially amplifying transcripts with lower GC content. And now we can actually design improvements to the smarter protocol to get around this problem. So there's lots of different metrics here. How can we start to draw some initial conclusions? Well, just looking at the number of green check marks, there were a couple of methods we were particularly interested in. In particular, as I mentioned, the RNase-H had a fantastic ribosomal depletion, good evenness of coverage, extremely high correlations with the gold standard. The only thing that was a little bit lower was the overall mapping rates, but this is because we were not enriching for polyadenylated RNA. We were also intrigued by the SMARTER protocol here, where we actually saw that everything looked quite nice, except for the correlations with the gold standard. But again, this is a general problem for low-input RNA-seq libraries. The last metric, which is, of course, extremely important, is the cost of each particular sample. The RNA-H method is not yet currently commercialized, so it was actually quite cheap for us to do on a per-sample basis. The other methods which come from commercial kits are significantly more expensive. So this got us particularly interested in a couple methods, but one thing we were thinking very carefully about is that in the context of low-quality RNA, we were sort of artificially fragmenting the RNA, but maybe that wasn't a very good representation of the really challenging RNA-seq samples that we truly want to look at, for example, the ones in the clinic. And so what we chose to do is we took those two methods that we were particularly interested in, along with total RNA sequencing, and we did follow-up experiments on 2 human samples. One was a direct biopsy from a human pancreas, in which there wasn't time to sufficiently or quickly isolate the RNA, so it was highly degraded. And the other was a very challenging FFPE sample from a human kidney, which was actually being used as part of the GTEx project. So we just wanted to see, do the trends that we had previously observed in our previous analysis continue to hold? And if we look at ribosomal depletion, again, we see extremely efficient depletion using RNase-H, and a lower but still high level of depletion for ribo0. And in general, we saw that the relative ordering of metrics was quite consistent between our artificially fragmented samples and our truly challenging clinical samples. Again, that we see relatively low but consistent levels of exonic mapping for all different samples, and relatively similar correlations with the gold standard. It's worth noting that all of these metrics are lower for true human samples than they were for our artificial fragments, but the relative ordering of the library seemed to be quite consistent. So to draw conclusions from this, we're actually pretty excited about this RNase-H protocol. We feel it's a very flexible and accurate protocol for a very targeted depletion of specific RNA molecules. Now, it's actually very worth pointing out that here we're using RNase-H to selectively deplete ribosomal transcripts, but we could actually deplete any transcript or set of transcripts of interest. All we have to do is design the appropriate and print the appropriate custom DNA oligos that are going to be complementary to our RNA of interest. So given the flexibility and the low cost of this method, this is something we're very excited about. The other conclusion that we drew from our study is that low-input RNA-seq is at least a feasible experiment. There's enormous noise in this data. What I'm showing you here is for the SMARTER approach, how do things look if we use 1 nanogram with SMARTER versus our gold standard RNA data set. So there's obviously enormous noise here in the data. But it's clear that there's actually quantitative value to this low-input RNA-seq, and we can successfully make a library with high mapping rates that we hadn't been able to do before. And we thought, well, if we can do this for 1 nanogram, which corresponds to about 100 single cells, could we actually go down to the individual single cell input? So I don't have time to tell you much about this today, but our work originally on low-input RNA-seq has really catalyzed a firestorm of single-cell RNA-seq analysis at the Broad Institute. Today we've actually sequenced more than 15,000 single cells, and we're going strong. Some of our initial work, which was published last year, and will come out again in a month or so, was actually on bone marrow dendritic cells. But we're increasingly moving to more and more towards patient samples. Samples from biopsies from a GBM, samples from human blood, anything we can do to actually show the value of the clinical value of single-cell RNA-seq. But I think perhaps one of the most exciting applications is something that Mark referred to very strongly yesterday, which is the ability to actually do bottom-up profiling of very complex tissues. So all of us are concerned about the context, the possibility of heterogeneity in our data. And traditionally, the way that we try to overcome this is to take a very complex tissue, for example, a lymph node, and divide it iteratively into more and more refined groups, for example, based on the expression of markers. So we can do a broad-based dissection into some rough groups based on one surface marker, and then a more and more iterative refinement with further markers and larger panels as we go on. But if we can do single-cell genomic profiling, we have the ability to sequence large numbers of single cells. Now, with the appropriate bioinformatic analyses, we can try to group these single cells together and actually build our understanding and characterization of these tissues from the bottom up. So I don't have too much time to tell you about this today, but perhaps at the second Microgenomics Symposium. And with that, I want to acknowledge my supervisor, Aviv Regev, and Joshua Levin, who really led this work, and then Shen Adiconis and Diego Borges Rivera, who did the primary bioinformatic and experimental analyses, and to also thank Ken Levac, Andy May, and Joe Shuga at Fluidigm, who we've collaborated on extensively on our single-cell work. Thanks very much.
Thank you very much for this very nice talk and very informative talk how to pre-process our samples. Questions, please.
Hi, thank you for this talk. It was very interesting. I'm product manager at Takara Clone Tech, so I have good news for you. We've just released the version 3 of the kit, and with a new enzyme inside, and it should overcome this problem of GC content. And that's so. Actually, in our data, we see that we have more coverage of some genes.
Thank you. Well, I think, but it's actually a good point also, because by doing these targeted analyses, and it's really, I don't show them all here, although they're in the supplements of the paper, if we can identify the classes of transcripts where the protocol is performing most poorly, it's a window into how to actually improve the protocol. And so that's actually been a valuable thing for us and obviously for you guys as well.
Further questions? One question from my side. These RNase H digest plus the DNase digest gives the best results. None of the other methods has also an additional DNase digest.
Yeah.
Is there an additional input of these DNase digests, do you think?
So we found that when you're looking at higher, so I should just mention that with the DNase and RNase digest comes the requirement for a cleanup, right? And so as a result, that's why we can't use RNase H for very, very low input to RNA-seq experiments, which is a shame because we'd obviously love to do that. What we found, and maybe people have different views on this, is that when we're dealing with a sufficient amount of RNA, here we are sequencing a microgram of RNA, though it is highly degraded, we don't see a huge effect of DNA contamination if we do a DNase cleanup as well. We still see a very similar percentage of reads that are mapping to introns. So actually, that's probably the best point to make too. Many of those reads that were in those striped bar plots are mapping to introns, not introgenic sequence. So we do think they correspond to immature RNAs and not genomic contamination.
Okay, thank you. So, further question? If not, we thank you again for this very interesting talk, and we go further on to the next talk. And the next talk is